So I'm going to talk about taxonomy of scelionid wasps associated with stink bugs. Most of the Gryon part of my talk had to do with the Grata Bug, which is not quite so relevant to New Zealand. So I removed some of that for brevity, but I'll talk a little bit about the Grata Bug parasitoids. This is a quote that I really like from an Australian author. Simply put, it is impossible to conduct meaningful ecological research without being able to accurately and repeatedly identify the species involved. So my job as a taxonomist is to make it possible to accurately and repeatedly identify species. In this case, the species that are relevant to biocontrol of BMSB. So it may seem obvious that biocontrol needs accurate identification, but it's something that should not be taken for granted. Science hinges on repeatability. There's a very high cost of incorrect identifications in biocontrol programs. And in the United States, a taxonomic name is needed before APHIS will allow the introduction of a biocontrol agent. So this is where being prepared taxonomically enables a very efficient response. First, we can select parasitoids for host range testing. Discovery and provenance of adventive populations. This is something that is really growing rapidly. In every circumstance that I've been involved in, the parasitoid that was under evaluation for release against a pentatomoid showed up before it was intentionally released. Also, if we want to concatenate information about host associations, which is very important for biocontrol, then the names have to be assigned appropriately. If one laboratory is using one name and another laboratory is using a different name for a species, then you can't connect the information that each of them is producing. It also enables us to make educated guesses about taxa of interest. So if we know that a particular genus attacks stink bugs, then that's an obvious place to begin looking. Collaboration is necessary and synergistic. The BMSB program in North America is a big wheel, and I am just one cog in that wheel. Molecular diagnostics, molecular phylogenetics, all of these things contribute to what I do as a taxonomist that enables me to contribute back to the greater whole. So these are the two critters, brown marmorated stink bug and Grata bug. This came out a few years ago, showing where BMSB is located. Obviously, it's present in Europe much more than we see in that particular map. And in the US, it was estimated to threaten $40 billion worth of agriculture. So the high cost of misidentification, well, we have these big numbers, and so a lot of people are working on this. And so one of the high costs is that when you have lots of publications coming out about a particular species, if a wrong name is inserted, all of a sudden we have a lot of papers with the wrong name. So I started with Kim in 2013 when he and a postdoc, Christine Dykhoff, were doing host range testing with two candidate biocontrol agents, which were operating under the names Trisulcus flavipes and Trisulcus hallimorphi. So you can see on Trisulcus flavipes those arced ridges on the front of the head. That's a diagnostic character that make this species very easy to identify, and it's also one of the most widespread in the Palearctic region. So I tracked down the holotype of that species, Trisulcus flavipes, and as you can see, it does not have those ridges on the head. So I tracked down a species that was treated as a junior synonym, and this one, Trisulcus cultratus, was the actual match to what they had in quarantine. And this happens because Trisulcus cultratus was erroneously treated as a synonym in 1968. So here's another example of a very high cost of misidentification. This means that one of the most common species in the Palearctic region in all of those collections is mislabeled since 1968. It also means that all the literature since 1968 uses the wrong name for this species. Trissolcus heliomorphi was described in this paper by Yang et al., which generally is a very nice paper. They gave important information about the biology, a description that was pretty reasonable for people who don't specialize in scelionids, but no primary types examined. And that is a very dangerous thing to do. And in Washington, where I was a postdoc, this is the place where the type of Trissolcus japonicus was housed. So the very first thing I did as a postdoc was sync the name Trissolcus heliomorphi because we wanted to inject the correct name into the literature as soon as possible. This is my former PhD advisor, Norm Johnson, and we have him to thank for a lot of the taxonomy that precedes what I've done. In North America in the 1980s, he did 3 revisions of Trissolcus species groups that cleaned up a lot of historical mess, it synonymized a lot of species, and it provided species concepts based on a wide sampling of specimens. That enabled us to very quickly put together a key to the biocontrol agents in the event that they were released in North America. We were also able to test our key on people who are non-taxonomists. So since 2012, there have been parasitoid identification workshops. This is an example of one of those, and so we could bring our key to people who are going to identify Trissolcus, who are non-experts, and find out where they had difficulties. If someone said, oh, I can't see this character, then we provide a better description or better images. That's very rare for a taxonomist to be able to test their product on the end users before publishing it. That came out essentially the same time that invasive Trissolcus was found in the United States. We found it in Maryland, and then within a year also on the West Coast. Last year, this is an example of the folks that attend the annual workshops. We've had one every year. I've been a part of them every year since 2013. We teach them how to identify parasitoids that come out of stink bug eggs, protocols for how to preserve those insects, how to make life easy for me. So I receive thousands of samples for identification every year. So if they can prepare them and send them correctly, they get their results faster and I spend less time on it. And that really, 10 seconds times 5,000 specimens is actually a lot of time. This is also a really good place to establish working relationships. So between me and the various people that are attending the workshops, and for them to interact with each other. I also like to emphasize collaboration. I think that the BMSB community works together really well in North America, and I like to promote that. This is our first workshop outside the United States. Kim and I organized this in Montpellier in association with EBCL and CBGP, which is an acronym for a French institution. I don't remember.
Population management, population biology lab, yeah, part of INRA.
Okay. And this is because BMSB is a growing problem in Europe. A good chunk of my time as a postdoc was spent revising Trissolcus of the Palearctic region. This started off as a revision of Trissolcus in Eastern Asia in order to make sure that we had the correct names for the Asian parasitoids. And it turns out that many of the species are found across this entire region. So there are some species that are found from Japan all the way to England. And this meant that I essentially had to do the entire landmass to make sure that I had correct names. This treats 50 species as junior synonyms, so more than half of the species names were things that had already been described. It required a lot of effort to examine primary types in Russia, in Vietnam, and a lot of curators in European collections contributed by sending me primary types. So essentially all of my taxonomic decisions are based on primary types, and I photographed all of those types and made them publicly available. And my intention here is to leave a trail of evidence in the term of photographs so that other people can corroborate or perhaps contradict my results. Taxonomy is an iterative process, so I'm not going to have all of the answers now, but I want to make it as easy as possible for people to correct any mistakes I made, which you're going to hear about. So there's one collection that I was not able to access as a postdoc, and that is the collection in Dublin, Ireland, which has the Walker collection. Walker was active in the 1830s. These specimens are very old and they don't want to mail them, understandably. So in 2018, I was able to visit this collection. And in particular, I was interested in Trissolcus belinus, because this is the oldest name in the genus Trissolcus, but it had not been well characterized. And there it is. It looks really good for being 180 years old. And it just so happened that I was able to work with Francesco, who's a taxonomist who I know in Europe, shortly after that meeting in France. I was visiting a collection in Genoa, and we met there, and so we set up a miniature laboratory in his hotel room, during which he said, I think that some of the species you treated as synonyms are actually valid species. And he had done this through breeding experiments and some molecular data. Now that's very convenient because our molecular analysis was just about to come out as well. And we found exactly what Francesco found, that one of the species that I had treated as a variable species was actually 4 separate species. And they come out up here. So this is one of the challenges, is that sometimes you have species that really are variable, and sometimes there are species that vary by very subtle characters. This species, the diagnostic character, is this row of 3 hairs on the first ladder of Turgay. So that's something that I had noticed it, I had seen it, it's in the description, but I wasn't willing to separate species just based on those 3 hairs. It turns out that they really are diagnostic. So this is where the iterative nature of taxonomy can be accelerated by making all of the images public. Francesco was able to see everything I had done. And so both of these papers were published together in a special issue last fall, where we have Francesco's work that clarifies the names, and I was able to contribute my images of those specimens from Ireland, so we could sort it out to the very beginning, and then update the names in our molecular pipeline. Now that Palearctic revision turned out to be particularly useful because Trissolcus japonicus showed up in Europe. And so did Trissolcus mitsukurii. And Trissolcus mitsukurii is known to extend from Japan all the way into Australia. And we don't know yet from where mitsukurii came, because we haven't matched the DNA to any populations in the Eastern Hemisphere. Now for Australasian Trissolcus, we again have Norman Johnson to thank, who revised the fauna in 1991, and this provides a solid starting point for additional studies. In particular, Trissolcus mitsukurii is important for the Australians because this species is known to occur there. This is a specimen that was in the Australian National Collection in Canberra, collected in 1930. Interestingly, the species was introduced in 1962, but it seems to already have been there, perhaps at just very low levels. This is the holotype of the species Ecleoides, which was described in 1914 from Queensland. This specimen is in the South Australian Museum. And this was treated as a junior synonym of Mitsukurii by Johnson in 1991. So we know that Mitsukurii at some level has been in Australia since at least 1914. So my goals during this trip with Australasian Trisulcus is to photograph all the holotypes. So I want to have that photographic evidence for every species name in case there's some updates to Norm's revision. There were some species where he said, this is a very highly variable species, I'm uncomfortable with the limits. So those are things that we want to revisit with molecular data. Morphological variation is something that has to be considered. This is Trisulcus japonicus that came out of a brown marmorated stink bug egg, and this is Trisulcus japonicus that came out of a podiasis egg. So not only are they smaller, but they're also darker in color. And this is the kind of information that's really useful to taxonomists that comes out of host range testing. What kind of variability do we see that's purely based on the host? We also want to characterize Australian Mitsukurii and have at least CO1 data for all of the Australasian species. Oh yeah, and then also I'm discussing some methods for conducting surveys with the Australian groups. So this is the distribution of Grata Bug. Here's an example of the kind of damage it does. This is rooibos in South Africa. All of these little brown things are Grata bugs. And they have this interesting behavior where they select a particular plant and they may ignore the plant next to it. And so this rooibos on the bottom there has been decimated, and the one just above it is essentially untouched. So there were some biocontrol agents being assessed for Grata bug, one of which was Trisulcus hyalinopenis. And from my work in the Palearctic region, I knew that it was found in places like Israel, Spain, the Middle East, Pakistan, places that are generally hot and dry. Then a friend of mine in Southern California sent me this single male specimen that was reared from a Grata bug egg, which is, fortunately we had characterized the biocontrol population that was in quarantine with DNA, so we could sequence the DNA from this and match it, because males can be a little bit variable. It's hard to make a strong statement like this is an introduced species based on a single male. But I subsequently examined specimens that were collected in people's backyards as part of a biodiversity survey in California and found that, in fact, Trisulcus hyalinopenis was rather widespread in Southern California, but nobody had looked. So one of the lessons here is that somebody's got to look at the parasitoids in the environment. There may be biocontrol agents that are already here. So let's say BMSB shows up. You may already have parasitoids here that attack it that nobody has looked at for 30 years. A little bit about new methods. This is a colleague in southern Mexico who I was put in touch with by a mutual friend who had reared some parasitoids out of Grata bug eggs. I'm just going to see if I can fix this screen here. Okay. And these are scelionid. in the genus Idris, which is a very easy to identify genus, although the species are very difficult. And all of Idris are parasitoids of spider eggs. It's a really big genus with about 1,000 species estimated worldwide. So I called my guru and said, Lubo, there are some Idris that supposedly came out of stink bug eggs. He said, no way. So I contacted my Mexican colleague and asked him about his protocol, and they had sifted out eggs from the soil and placed 1 egg per petri dish. So it seems that there was no chance of contamination. And he had kept the eggs. So this is a healthy, unparasitized egg, and this is the empty shell of an egg from which the parasitoids emerged. For people who work in Platygastroid taxonomy or systematics, it's very hard to believe that Idris would attack a stink bug egg, so I knew that we needed some kind of supporting data. And my colleague Tara Garapi in Canada has this method that Kim alluded to earlier, in which she can take an empty parasitized egg, sequence the host DNA and the parasitoid DNA, and match it to Koan databases if they're available. And so this is exactly what we did. Refugio sent her one of the empty eggs. I sent her the parasitoids from Refugio. She was able to sequence Grata Bug and Idris DNA from that empty eggshell, which was a 100% match to this adult Idris. And this is a great example of when an exotic creature moves into a new habitat, that there may be host associations that nobody's ever thought of before. I don't think that anyone expected a species of Idris to attack a stink bug egg. It's also a nice example of collaboration. I particularly like it because it has a Canadian, someone from the US, and a Mexican working together with each of their respective specialties. Okay, so here are a lot of people to thank.